Bismillahirrahmanirrahim. It's chapter number 17, Selection and Evolution. What is genetic drift? Change in allele frequency due to a chance event. For example, we have the petals, blue and red petals, 5 and 5 ratio. And by chance, some, you can say the petals have been killed accidentally, and the remaining red beetles will reproduce, and in the next generation the ratio will change 10 to, because the beetles which have been survived, uh, they will reproduce and they will carry their genes forward. What is the founder effect? A few individuals from a population start a new population with a different allele frequency than the original population. For example, this is example of the founder effect, that a few alleles from this population, they start, for example, this is a population. The red beetles flew away, or there is a process of migration from this population, and this red beetle will move to another island and another area, and so only these red beetles will survive or will increase their generation. So this is a few individuals from a population start a new population with a different allele frequency than the original. Original carries the yellow and red, whereas the new one carry only red population. What is the gene pool? Populations are made up of the members of the same species that interbreed, and variation naturally occurs among the genes within a population, and the collection of all the genes and the various alternate or allelic forms of those genes within a population is called the gene pool. So we can say that all the kind of genes and alleles in a population, these are called the gene pool. What is allelic frequency? The frequency of any given allele in a population relative to all the other alleles at the same locus is known as the allele frequency, that is equal to number of copies of a particular allele in a population divided by total number of all the alleles for that gene in a population. Example of allele frequency: how often any form of a gene shows up in a population over several generations, and the number of copies of a particular allele divided by the number of copies of all the alleles at the genetic place in a population. For example, this is homozygous dominant, this is heterozygous, and this is homozygous recessive. Now, what is the Hardy-Weinberg principle? Frequency of homozygous dominant genotype plus frequency of heterozygous genotype plus frequency of homozygous recessive genotype is equal to 1. Now, this is the Hardy-Weinberg principle. It states that frequencies of alleles and the genotypes in a population's gene pool remain constant over the generations unless acted upon by the agents other than sexual recombination. For example, the migration or the non-random mating and the other factors also. So we are going to discuss with the help of an example. We are taking the example of the wildflower. The wildflower, they have two colors in their phenotype. One is pink, other is white. The pink allele of that white color flower is dominant over the white allele. We denote the capital A to pink and small a to white, as white is a recessive form. Suppose we have taken the population of 500 plants. So we are considering, for example, that white, of course, the white color can express itself only when it is the homozygous recessive. The white one are 20 in number. The pink ones are, for example, 480 in number, out of which 320 are homozygous dominant and 160 are in heterozygous condition. So as diploid organisms, individuals are present. So 100 copies of genes as alleles are present in a population of 500 plant, 500 into 2. Then the dominant allele is, for example, it's carrying the, that is the capital A, carrying the 320 as the dominant form. So capital A is present. So we will multiply 320 into 2. This will make 640. And in the homozygous dominant condition, and in the heterozygous condition, that is capital A small a. So only a single allele is present representing the heterozygous condition. So 160 is there. So 160 plus 640, they will make 800 alleles. So 800 alleles of only are only present for the pink flower. So we are considering the ratio of homozygous or heterozygous out of 800 pink flowers. So frequency of allele A, capital A, is the number of that allele out of total. That is 640. 640 in case of the dominant homozygous, that is 640 out of total. Total mean heterozygous and homozygous, that is 800. It is actually 0.8 or 80%. Then the frequency of allele small a was 20%, and that is 0.2 because that is homozygous recessive. Capital A means 0.8 into 0.8, it will make 0.64 or 64%, or we can say 320 out of 500 will make 0.64. In case of the heterozygous condition, capital A small a, 0.32, as the heterozygous contain 160 genes out of 500. Small a is 0.04, as the homozygous recessive, they contain 20 genes out of 500. Now, according to Hardy-Weinberg theorem, P plus Q square is equal to P square plus 2PQ plus Q square is equal to 1. It should be 1. So by putting the values, we are taking P square as the homozygous dominant. 2PQ mean P is the dominant allele, Q is the recessive allele, and Q square, it shows the homozygous recessive form. So we will, when we will put the ratios, 0.64 for the P square and 2 into PQ, this one, and plus for Q square we will take the values, that is small a homozygous recessive, that will be equal to 1. So we can take the value, we can measure the value. If P plus Q is equal to 1, then 1 plus P is equal to Q, and 1 minus Q, 1 minus P is equal to Q, and 1 minus Q is equal to P. So that we can determine, or we can find out the values of the, you can say the frequencies of alleles and genotypes in the population by using this formula. And we can also observe that what is the difference between the, you can say, our expected values and our observed values. Now, the basic assumptions of the Hardy-Weinberg principle that all the phenotypes they are equally fitness, no natural selection is there, no mutation, no immigration, no emigration, no genetic drift, and no assortative mating should be there. So this is the ancestral population. This is the Hardy-Weinberg equilibrium, and events, random mating, no migration, genetic drift, mutation, or natural selection by the passage of time. Later population, of course, that is results in the allelic frequencies do not change. Of course, at least one of these factors will be acting on a population in the wild. Otherwise, this doesn't happen normally. The importance of the Hardy-Weinberg theorem is enables to compare the population's actual genetic structure over time, and if genotype frequencies differ from those we would expect under the equilibrium, we can assume that one or more of the model's assumptions are being violated. So with the help of the you can say the Hardy-Weinberg theorem, we can determine the gene frequencies or allele frequencies in a given population.